Hi, I'm Beth Walczak. I recently completed my PhD training with Gary Hammer at the University of Michigan. Today I'll be giving you the highlights of our molecular endocrinology paper stemming from that research. So the Hammer lab is interested in how adrenocortical stem and progenitor cells are regulated by paracrine signaling pathways, such as the canonical Wnt signaling pathway. Beta-catenin, which is the transcriptional effector of this pathway, is critical for proper adrenocortical development, postnatal homeostasis, and is involved in tumorigenesis of the gland. So we wanted to investigate whether Wnt-responsive cells in the adrenal cortex are comprised of a proliferating progenitor cell population. But what we found is that Wnt-responsive cells actually infrequently colocalize with actively proliferating cells, and this population is heterogeneous. It is composed of undifferentiated adrenocortical progenitors, as well as differentiated steroidogenic cells of the aldosterone-producing zona glomerulosa, but not the corticosterone-producing zona fasciculata. So we speculated that perhaps Wnt signaling prevents the differentiation of adrenocortical progenitors into fasciculata cells. So using an adrenocortical cell model, we set out to test whether induction of beta-catenin activity represses fasciculata steroidogenesis as a readout for differentiation. When we pharmacologically stimulated beta-catenin activity, we observed repression of corticosterone production due to reduction of the transcription factor SF1, both its expression and occupancy at steroidogenic promoters, resulting in reduced expression of steroidogenic genes. So next we sought to determine adrenocortical Wnt targets that could participate in this phenomena. We identified genes whose expression is enriched in the Wnt-responsive population in vivo and stimulated by beta-catenin in our cell model. One candidate gene was CCDC80, which encodes a secreted protein whose function is unknown in the adrenal cortex. So CCDC80 is expressed in the subcapsular Wnt-responsive region of the cortex, and it requires intact TCF binding sites for stimulation of its expression by beta-catenin. In conditioned media experiments, we found that treatment of cells with CCDC80 blunts steroidogenic gene expression and corticosterone production, phenocopying the effects we see with beta-catenin on fasciculata steroidogenesis. Collectively, these data suggest that Wnt signaling, through actions exerted by beta-catenin and CCDC80, represses fasciculata steroidogenesis. Combined with a recent report implicating Wnt signaling in glomerulosa steroidogenesis, a model emerges in which Wnt signaling in the adrenal cortex facilitates the differentiation of an adrenocortical progenitor into a glomerulosa cell while preventing the differentiation into a fasciculata cell. Please read our paper for more details. Thank you for watching.